In a single week, the tech world watched an AI model hack another firm, while a different system broke out of its isolated test environment. Against that backdrop of digital guardrails snapping, a specific headline started trending. Artificial intelligence had just designed a working virus. The response was immediate, filling social feeds with doomsday scenarios and bioweapon theories. However, the panic overlooks two key details: the specific nature of the experiment and the fact that this breakthrough actually happened 11 months ago. The current news cycle was triggered when research from Stanford University and the Arc Institute cleared formal peer review and was published in Science. The team didn't build a digital plague. Instead, the researchers used the experiment to document a specific security gap. They successfully demonstrated that even if an AI model were used to design a dangerous sequence, the systems currently in place to block those sequences from being physically printed are largely voluntary. The Stanford team used the phage as a tool to white hat hack the biological supply chain, revealing a legal blind spot that exists today, long before any AI-driven pathogen could be synthesized. To understand the actual risk, we have to look at what the AI built. The underlying generative breakthrough happened back in September 2025, when the team released their initial findings on a preprint server. The formal publication is new, but the science is nearly a year old. The engine driving this research is an AI model called Evo2. Instead of training on libraries of human text, Evo2 was trained on millions of whole genomes. It reads and predicts sequences of DNA the same way a language model predicts the next word in a sentence. The researchers didn't ask the model to invent a pathogen from scratch. They gave it a highly specific template, a tiny 11-gene virus known as phage174. This is a bacteriophage, a virus that hunts bacteria, not people. This specific bacteriophage only targets E. coli bacteria. Its genome contains just 11 genes, a fraction of the complexity found in human pathogens, and poses zero infection risk to humans, animals, or plants. The team deliberately excluded human-infecting sequences from Evo2's training data. Because of those specific guardrails, the output is a controlled proof of concept, incapable of infecting a human cell. Viral posts imply an AI system casually cranking out flawless viruses on command. The hard data from the Stanford study shows the technology's current limits. This chart tracks the experiment's success rate. The AI generated 302 candidate phage genomes. Only 285 were capable of forming a full sequence. Of those 285, just 16 worked in a lab to inhibit E. coli, a 5% success rate. That success rate translates to a functional clinical tool. When the researchers combined those 16 successful phages into a single cocktail, it rapidly overcame bacterial resistance across 3 separate strains of E. coli. For now, this technology functions as a brute force therapeutic research assistant, churning through trial and error to fight drug-resistant bacteria. It is a tool for lab bench discovery, not a turnkey weapon. While the viruses from this specific test are harmless to humans, the biosecurity community is focused on a different problem. Alongside the Stanford paper, Johns Hopkins specialists published a companion editorial in Science pointing directly at a gap in the law. The concern stems from the fact that Evo2 is open source. The model is available to anyone with an internet connection and the computing power to run it. There is currently no technical barrier preventing a motivated actor from taking a similar open-source architecture and training it on publicly available genomic data for human pathogens. But generating digital code is only the first step. To create a physical threat, a person has to turn that digital file into physical material. They must send the sequence to a commercial laboratory, like the machinery shown here, to have the synthetic DNA custom printed and shipped. And this is exactly where the governance fails. Currently, no US law requires commercial DNA printing companies to verify the genetic sequences they manufacture, nor do they have to verify the identity of the customer. The screening system today is entirely voluntary. Generative AI can now produce functional blueprints for living DNA. However, the legal screening needed to stop those digital files from being synthesized into physical material has not been built.